afternoon, everyone. Yeah, thanks. My name is Jung-Eun Choi from EWHA Women's University, and I'm a student from College of Pharmacy. Under the group theme of Science Unlocking Future, I'll briefly introduce my research about stress proteomics, and then I'm going to discuss what we can do with those stress to make our future more enjoyable. So the proteomics is a systematic study of protein expression profiles in certain conditions. And I focus much more on the post-translational modifications of each protein I identified in my study. So the post-translational modifications, PTMs, is a molecular remodeling of a protein which occurs after the protein synthesis process. And to study these PTMs is really important because these modifications can change the function of protein, and finally it can regulate the cellular function in different ways. When we are exposed to chronic stress, oxidative stress may increase in your brain, and it can cause, it can be one of the causes of major depressive, major depressive disorder. And to study the molecular changes occurred in depressed brain, I used this special mouse model, which is chronic restraint stress model. And briefly, mouse is restrained in a narrow test tube for 2 hours a day and for consecutive 9 or 14 days. And I used the hippocampus of this mouse to study the proteome changes of the depression model mouse. And it is also known that oxidative stress is increased in this mouse brain is also known. First of all, proteins from control and stress mouse hippocampus were separated using 2D page based on their size and based on their electrical property and their size. And I found that 63 proteins were changed after chronic stress, and I identified that protein spots using mass spectrometry. And it was found that the proteins involved in neuroplasticity, metabolic process, and protein aggregation were changed after chronic stress. I continued my further research on UCHL1, which showed the most dramatic change after chronic restraint stress. And these 4 spots, spots 1 to 4, are all UCHL1 spots. And it seemed, interestingly, spot 1 disappeared by the stress treatment, and spot 4 increased by the stress treatment. And it just seems like those spots are shifted by the chronic stress, like this. And I studied which is the differences between those 4 spots. And using mass spectrometry, I found that UCHL1 spots are diversely oxidized at their cysteine residues. Especially cysteine 90 and cysteine 152. And since I found that UCHL1 is diversely oxidized in the stress brain, I investigated the effect of UCHL1 oxidation in cellular oxidative stress model. The cell line I used was HT22 hippocampal neuronal cells, and oxidative stress was induced by treating hydrogen peroxide to those cells. And I'm not going to describe a very detailed way about these biochemistry things, but only wild-type UCHL1, which contained cysteine residue with its unoxidated states could exert protect effect against oxidative stress, and these cysteine mutants, which cannot be oxidized by oxidative stress, could not protect cells from oxidative stress. And protein oxidation is often regarded as a bad thing, but in this case, in my study, oxidized UCHL1 protected neuronal cells from oxidative stress. So is stress really harmful to our health? Even though there are so many studies describe that stress can cause harmful effects to our health, but there are another study reports that says moderate stress, which is not severe enough to cause death. It is important. Yeah, can help prolong our life. Can help prolong the lifespan of C. elegans. And so the message I want to convey from my research is that stress is present everywhere, but we also already have self-defense system such as UCHL1 I show you, and we already have those self-defense system, and even some stress can prolong our lifespan in a positive way. Yeah, and we're facing the fourth industrial revolution. And when I searched it, yeah, again, yeah. When I searched it on the internet to find the definition of this concept, and I was so excited to see that economists pointed us to be the future leader. Yeah, let's see. It's characterized by a fusion of technologies that is blurring the lines between the physical, digital, and biological spheres. So that means us, right? Yeah. And in the next generation, the fusion science will be really focused, and the value of human nature will be more increased. So I guess the health issues and maybe leisure life contents will be more emphasized, and rather than the simple labor, which can be just replaced by the robots or kind of things. And here is the Marvel hero, Iron Man. And in my opinion, science fiction always reflects our future perspectives. So let's just take a few seconds to guess which science fields are related to this Iron Man. Yeah. Everything, I guess. And what I got here is energy engineering, yeah, from the core, and robotics, of course, biotechnology to get to check vital of Robert Downey Jr. Computer science, the ethics is for where to use our power, yeah. And maybe urban planning. I put the urban planning for Jung because urban planners should fix all the destroys, yeah, reconstruct destroyed cities after the war, yeah. And material science and aerospace engineering, I guess. Yeah, so it's all of our science fusion in the one character. Okay, so it's my conclusion here. So stress is inevitable, but it is up to us to how to deal with that stress. And maybe UCHL1 can help your hippocampus safe. And as a young scientist, we can make the future by ourselves, and let's keep challenging and to face the wave of change with flexibility. And networking is important. And the last thing, I guess it's really important. Let's find happiness in our personal life. And these things are makes my life happy. And my family here, and short walk after lunch with my lab mates, and my little garden in the balcony. Yeah. And thanks for listening.
Good afternoon, everybody. So today I'm going to talk to you a little bit about my research and how it impacts the future. And the title is RNA Regulates Gene Expression via Triple Helix. So the reason I got into research is because I think it's important for us to make our world and humanity better. So I like to study things that are happening in the human body. So this is my family. I will show them every chance I get. So in the middle here are my parents, and this is me. And then this is my older brother, and then there are 9 of us, and this is my youngest brother, if you couldn't guess. So they have given me a lot of support and they have made my life better, so good. And I grew up in a small town in Ohio, so that's just a little bit about me and my passions, I guess. So we're gonna go back to high school, back to the central dogma of molecular biology. Most of you probably know this or have heard this before, even if you don't remember. So in our body is DNA, and the DNA codes for everything, who we are and what we become. And the way that it does this is that our body uses the DNA as a template to make RNA, and then our body uses RNA as a template to make proteins. And this is what most people know about RNA. It's just kind of this middleman that does, well, nothing else other than that. So, but it turns out only about 2% of the RNA that our body makes is actually used to make proteins. So what is the other majority of our RNA doing? So one group of these are called long non-coding RNAs. This is where my lab focuses. So there are over 100,000 known long non-coding RNAs in the human cells. So that's a lot. And they have been implicated in several biological functions, in different disease states. And this picture here, there's a lot going on. You don't really need to know what's going on. It's just so that you realize that there's a lot going on. So all the red in the picture is long non-coding RNAs that were thought before to do nothing, and well, they're doing a lot in the cell. My work focuses here, right in the middle, where long non-coding RNA is binding to the genomic DNA that codes for everything in order to upregulate or downregulate transcription of certain genes. So these may look familiar to you. This, for DNA, we have we have 4 nucleotide bases. A binds to T, and G binds to C, and they form this ladder of binding together with a lot of these stacked on top of each other. So my science proposes that there's long non-coding RNA that comes and binds to the genomic DNA, and if you like diagrams here, we're going to zoom in just on one of these, and it looks like this. So they bind hydrogen bonds, the same as the double-strand binds. The ones I show here are well known to form. So the UAT is called a base triple, and the CGC base triple are well known to form. But you can imagine that maybe other bases could be changed in. And so that's what my research focuses on. So what happens if you put an A here instead of a U? Does this triple helix still form? So this is a crystal structure of, it's called mallet 1, long non-coding RNA. And this is a triple helix that's very well known and studied in the field. So, but this is just to show you that the double helix goes around and the third strand wraps around it. So what do? I mix double-stranded DNA with RNA, let it sit around in a test tube for a little while, and then I run it on a gel. So if I have no RNA in the gel, or in my sample, then I have this band here. This is showing that, so this band indicates where the two DNA bind to each other. But if I add a lot of RNA up here at 1,000 nanomolars, we don't see any more of the double-stranded, and it shifts up to the triple-stranded. So this is what I do for a living. It's a good time. So if, as I said before, you can change out the bases, and if you do that, you can see that different bases have different stabilities, so, or how well they bind to each other. So this graph shows 16 different base triples. These two are the well-known ones that I pointed out at the beginning. So they're the tallest. That's good. These three down at the bottom that I boxed off, they showed no binding, so I couldn't get the RNA to bind to the DNA. And then these 7 are 7 non-canonical or not well-established base triples that show at least 50% of the stability of the normal ones. So how is this unlocking the future? Well, it will help us figure out what these long non-coding RNAs are doing. It can help with diagnostics. So there are algorithms that exist that predict where long non-coding RNAs are binding in the genome in order to regulate transcription. But the problem is they only use the 2 that I mentioned before. But my research shows that there may be a lot more, more things to consider. So yeah, that's about it. I started with the picture, and I'm going to end with the picture. So this is my lab. We're pretty new, small lab. I'm my PI's first, so that's exciting and terrifying. So yeah, so thank you.
Hi, good afternoon, everyone. I'm Li Lu, and I'm from Yangzhou University. Today, I'm going to talk about my research on ordered porous membrane assembled polystyrene microspheres as electrochemical immunosensors. Yangzhou is a very famous tourist city in China, and this is the Slender West Lake. It's also a very famous lake in China. And this is my university. Very beautiful and traditional. And yeah, here's my contents. I'm gonna talk about my research in these 4 points. First, ordered porous films. As we all know, the ordered polymer films have attracted many interests because of its potential applications in field of separation, and biosensors and templates and so forth. Here is some methods to fabricate these films. However, the lithography needs high equipment cost and very time consuming, and the emulsion droplets cannot control the size very well. So we chose the brace figure method. We called it BF method because it's very simple and very cheap and very easy. And next is the mechanism of BF method. The base of this method is the water droplets self-assembling. When the polymer solution casting on a substrate in a humid air flow, with the solvent evaporation decreased the surface temperature, and then the water condensed on the surface, and then after some time the water droplets are gathered in ordered under the surface tension gradient convections and then the water droplet organization. After complete solvent evaporation, this water droplets united like ordered microporous polymer films. Looks like a honeycomb pattern. And here is the equipment flow chart to prepare these porous films. And these gas flow meters to control the speed of the air, and we also there is the water to control the temperature, and then we can get different size of the holes, and we use this PSBPA in chloroform solution. And here is some SEM images of these porous films at different concentrations of complexes. We can see under different magnifications, it shows very regular ordered and the holes, the size between 500 nanometers to 4 micrometers. And then next is porous membrane self-assembled microspheres. We use polystyrene microspheres. As we can say, we use these two different ways to disperse the microspheres. It shows different states, and we use this because it's more. The film's surface is more smooth and more stable. And here is the SEM pictures of this self-assembled. It's also very, very regularly, and the surface not broken. And then we also choose the silicon oxide, and in order dispersion on these films, it shows all the balls are separated on the film surface, not in these holes, just different from these ones. And then we need to find the application of these materials. So we choose label-free electrochemical immunosensors. So, as we all know, the biomarkers, also known as Tudor markers, are very important diagnostic basis for clinical cancer. So we choose this CA99 to test pancreatic cancer. But now the most methods to detect cancer is radioimmunoassay and enemy-linked immunosorbent assay. They have so many disadvantages. They require labeling of the antibodies or antigens, and it's complex and very time-consuming and very expensive. And they are very sensitive, although. And However, we chose this label-free immunosensors. It's very sensitive, very low cost, and very easy of preparations. And it has a very fast analytical time. So here is the basic working principle of these immunosensors. It's just like a sandwich type. And first self-assembly, and then target input, and then it worked, and cycle, and last signal output. So here's the basic experimental flowchart of these immunosensors. You can see it's the condition is very simple, and it won't take too much time. And then we use ELIS to test the data, and because the ELIS is often used for monitoring the interfacial properties of modified electrodes and employed for the analysis of electrochemical process associated with electroactive substances. So when the electrode coated with the antibody protein is immersed in the corresponding antigen solution to be tested, the antigen and antibody undergoes specific immunoreactions on the electrode surface to form an insulating or semi-insulating antigen-antibody composite film. In a certain range, we can by plotting the standard curve, the concentration of the unknown analyte can be calculated, and then we can know the limit, the limit of the detection. And as these pictures showed, here is one of our test data. It's very it's raised step by step, so it means this immunosensors is working. So the last is impacts on the future. We want to develop a new type of electrochemical sensor for disease detection and early prevention. And then this technology is based on the specific binding of various antigens and antibodies. So it has a very range of application. It can test more and more cancer disease. And this impedance electrochemical technology used in this research can also be greatly developed in the field of environmental and food testing. Thank you for listening.
Hello, everybody. How's it going? You guys all look like you have enough snacks. Good. My name is Hannah BOSSI, and I went to my undergraduate education at Colby College, which is in Maine, same as Alicia. And for my graduate education, I'll be pursuing a PhD in physics at Yale University this fall. And I'll be talking about space, time, and quantum gravity. And I know that those three words seem really complicated, but we'll unpack them as we go. So just to give a little bit of an outline, my topic is science unlocking the future, so I thought it would be only natural to talk about how science of the past impacts science of the present. And then I'll move on and talk about how the science of the present, specifically the science that I work on, is going to impact science of the future. So we're going to talk about a little bit of a story, and this is very biased towards physics because I am a physicist. So we start with Newton's laws, and Newton was actually originally a philosopher, so people like him kind of worked out how heavenly bodies moved and how they interacted. And a lot of people thought he was crazy to quantify it. They called it experimental philosophy, and he got a lot of judgment for quantifying the universe. But we now see that even though he was pursuing something seemingly meaningless and quantifiable, the applications of Newton's laws are endless. They permeate our society, and we surely would not be where we are without them. And now let's go to another often thought as meaningless science, general relativity. So general relativity is very complicated. When it was developed, only 3 people supposedly on the Earth knew how it worked. And that's after reading all of the papers. And general relativity was often thought of as like a science that never affects everyday people. But we often find as this technology develops that it really does. And general relativity leads to development of the GPS. And where would we have been on this whole trip without GPS? And now let's move to Cern and the Large Hadron Collider. So Cern was another thing that was often disputed as to the funding. People thought that Cern and developing these very expensive tools to probe the early universe weren't as necessary to society today. And it actually turns out that technologies developed here are something that we use every day. In fact, I'd be willing to bet that some of you are using it right now. And so 2 very important technologies came from this. So the World Wide Web and touchscreens. So we see from these 3 examples that even theoretical physics that doesn't really have a clear connection to society and technology really does impact science and technology for future generations. And so now let's talk a little bit about my work. So a lot of you have already seen this slide of my small seminar. And what I work on is quantum gravity. And basically, physics has many different types that act over many different scales. So one instance, we have quantum mechanics over here. And quantum mechanics describes physics of really small things. And then we have general relativity, and that describes physics of really big things. And as physicists, we like to make things way simpler than they actually are. So really, we would like these 2 to merge into one grand unified theory. And that's kind of what I'm working on today. And I modified general relativity to contribute to a grand unified theory. But first, we need to do some background before we talk about this theory of quantum gravity. So we're going to talk about Lorenz symmetry. And physics is based on symmetry as a discipline. It's very important to understand this. So Lorenz symmetry is the invariance of the laws of physics under Lorenz transformations. And that sounds way more complicated than it actually is. So let's take a look at this table right here, for example. So I see this table from my perspective because I'm looking at it from the stage. But each of you have an individual perspective on what that table looks like for you. But the laws of physics don't change based on where we're standing in this room. That table is still motionless. I could still quantify all of the different aspects of that table, and it doesn't change what's actually going on. And these are the formulas for Lorenz transformations right here. And they tell us something really important. So for example, we see that this quantity x and this quantity t are subtracted from one another. And we all know as scientists that we can't subtract apples and oranges. It just doesn't work. So when we're subtracting things, we're saying that they're fundamentally the same thing. So let me repeat that. Space and time here are fundamentally the same. And the way the English language works, and many languages do this, they put time in boxes. They put it in a day, a minute, a year, a month. They're all putting it into boxes, but it doesn't actually work like that. So space we can understand. We can move through it. We can manipulate it. We guide it to how we want it to be. And time also works in this way, but we don't see it that way because of the way we talk about it. But space and time is really integrated into this one single 4-dimensional fabric that makes up our universe called space-time. And so this is really important to when we talk about general relativity. So general relativity is a geometric theory of gravitation that was developed by Albert Einstein with, I think, a little bit of help from his wife in 1915. And so general relativity, as all the physicists in the room would attest to, is incredibly complicated. But basically what it says is that massive objects distort space-time, and we feel this as gravity. So I could drop this clicker right now. I'm not going to, because then I'd just have to pick it up again. But if I did, it would fall to the Earth, and we know that it would because it's attracted by Earth's gravity. But what does this actually mean? So let's say this larger ball in the middle is the Earth, and this smaller thing is the clicker right here. And so we know that if I let this small marble flow freely, it'll go into that well and be attracted to that larger object like the Earth. And that's the same thing as saying when I drop my clicker, it will naturally fall to the ground. So we see here that gravity is not really a force in the way that we're always taught that it is. And it's actually just a distortion of space-time and objects moving freely within that space-time. But if you recall what I said a few minutes earlier, general relativity is wrong. So this is really troubling for physicists. We love general relativity. It works so well. Our GPSs hardly ever fail, so how could this be wrong? And the reason why is that it's non-renormalizable. And the mathematics behind what that means doesn't really matter. But it basically means that at very high energies, we can't predict what will happen. And mathematically, that looks like a lot of infinities popping up where they shouldn't be, and it all gets very complicated. But in the infrared region, we know that general relativity works well because of all the different reasons that I specified before. We know that our GPSs and things are accurate, and these are just one of many different tests of general relativity. And at the UV region, we see that this loss of theoretical control makes it incredibly hard to predict, and a lot of things don't make sense. So something has to be wrong. So when we're looking for a quantum theory of gravity, we need to make sure that this renormalizability is fixed. And that's what I work on. I study a quantum theory of gravity called Hobbit Gravity. And the basic concept is that it's a power-counting renormalizable theory of gravity. And that all sounds complicated, and it doesn't really matter what that means. But the main point is that there's an anisotropic scaling of space-time. And that means that space and time scale differently. So mathematically, it's represented like this. But we see that there's one big consequence. When we scale space and time differently, they're not the same, essentially. If I step one way in a spatial direction, that's like taking 4 steps in a temporal direction. That can't happen, and that breaks that fundamental symmetry. So for example, if I was moving around this table, it would scale and stretch as I moved. And that doesn't make sense. I should be able to just walk around the table. But in the high energy limit, this might actually be how the universe works. And so here we have to really think about the physical consequences. So let's say we have 2 different foliations. And again, that word is very complicated, but what it actually means is just a way of space-time. And so we see, let's consider that this vertical axis is a time axis, and then these sheets are 3D space. Now, I know they're 2D, but we have no visual representation of 4 dimensions, so it kind of has to be this way. And in general relativity, these 2 foliations are the same. I would just turn my head and all the math would be different and it would just transfer over. But Hobbit gravity has a preferred foliation, as I was saying before. And so this means that space and time may have a preferred direction in the universe. And the amount of technology that this could unlock is unimaginable, and we really have a limited idea. But in conclusion, we know that science of the past is deeply embedded in our everyday lives in ways that we didn't always expect, even from a theoretical physics. And we also know that present science will impact the future in new and exciting ways. And this also includes Hobbit gravity as a theory of quantum gravity that treats space and time differently. And ultimately, as we all study science, the future is ours. It sounds cheesy, but we do know that since science of the past impacts science of the present, we do know that we'll undoubtedly all impact science of the future. Thank you.
Good afternoon, everyone. My name is Nicole, and my major is physics in Seoul National University. Today, I want to talk about superconductivity and how it will change our future. So this is the content I would like to deliver to you today. Firstly, I'd like to introduce what is condensed matter physics and what is superconductivity. And secondly, I'd like to talk about how this superconductivity will unlock the future and some limitations and efforts to achieve it. So actually, there are a lot of branches of physics, like Rnas did, nuclear and particle physics, and there are like atomic, molecular, and optical physics. And condensed matter physics is one of branches of physics. This is for describing behavior of condensed matter physics in terms of physical laws. We can use a lot of physical tools to describe it. For example, if you want to see really, really small world, we can use quantum mechanics. And if you are interested in electron-electron interactions, we can use electromagnetism. And if you are interested in a number of particles, we can use statistic mechanics. So what is condensed matter? Actually, it is really easy. You all know about what is solid, liquid, and gas, right? So this circle is atom. So in liquid and gas phase, atoms continuously move, yes. But in solid, atoms are at fixed point, which makes this kind of lattice, repeated structure. So this gives some interesting phenomena, and we are interested in to figure out what phenomena it comes from. We can classify condensed matter by a lot of ways. For example, by structure, whether it is cubic or tetragonal and so on, or by chemistry, what atoms consist of, or last and the most importantly, resistivity. This is the mostly used one. As you know, all materials have resistivity. So we can classify condensed matter by insulator, or semiconductor, or metal. Then what is superconductivity? As I mentioned before, all materials have its resistivity. So this mic or this desk have resistivity. I think this mic is conductor, so it has lower resistivity, which means electron can go freely inside of this material. But this desk is insulator, have higher resistivity, so electron cannot go freely inside of it. Here you can see normal material have finite resistivity, but superconductor have zero resistivity under some critical temperature. So it is really special. Because inside of superconductivity, electron can go freely forever. It can produce a magnetic field like forever. Then how can this unlock the future? Because it can generate persistent magnetic field, we can use it for magnetic levitation train or small and really cheap MRI. And also we can use it for computing. Actually nowadays we are using bit for computing, which have only two states, 0 and 1. But superconducting states have infinite states, so we can use it for quantum computing, which will make computing much, much more faster. So a lot of people think this superconductivity will realize quantum computing. But why are you not using this superconductivity these days? Actually, there is really big problem limitation. I will talk about this limitation and efforts to overcome this. Critical temperature is too low. The highest critical temperature so far is 135 Kelvin, which is under minus 100 degrees Celsius. This shows year and the critical temperature depending on the material. As you can see here, it keeps increasing gradually, but still it's too low. So we need to make room temperature superconductivity, but it is really hard because we don't know a general rule about superconductivity. So a lot of scientists are working on to figure out where this superconductivity comes from. Like, there are a lot of theories and experiments, and I am one of experimentalists, so I am working with finding out whether the theory is right or wrong. So in the future, I hope we can make room temperature superconductor and unlock the future. Thank you.
Good afternoon, everyone. My name is Tricia CHAFFEY. I am a recent graduate from Mount Holyoke College, and I will be beginning my PhD studies in computer science at the University of Southern California in the fall. Now, today I'm going to be talking about social formations in an education setting between a student and a robot. But before I get into detail about this, I'd like to pose a scenario for all of you to think about. I think it will hit a little close to home for some of us. Now, you're working on an assignment or a project, and the problems are very difficult. As time goes on, the problems seem to get harder and harder, and eventually you hit a wall. You reach for your phone thinking, I'll text my friend, maybe I can bounce some ideas off her. But you look at the time and you realize it's too late. She's asleep, and you're alone. But what if you didn't have to be alone? At Mount Holyoke College, we seek to address this problem by designing a robotic learning companion. Now, within the context of this ongoing project, I conducted research into the social formations, and I will get into detail in just a moment. So, what are social formations? These stances are the positions we take with one another when we are involved in a conversation. For this project, we looked at two stances between a human and a robot, side by side and face to face. The side by side stance and face to face stance were selected because we feel that in a tutoring scenario, these are the most commonly adopted positions. Now, at Mount Holyoke College, I should back up a bit. At Mount Holyoke College, with our ongoing project, the robotic learning companion that we're designing is not a traditional tutor in the sense of the robot teaching facts to the student and the student taking notes. Rather, we ask our students to instead teach these concepts to the robot because we feel that students learn best when they can teach the material to another individual. Now, why do we think that humans are aware of where a robot is positioned around them? Well, previous studies have found that robots are capable of both explicitly and implicitly reconfiguring the social scene. This means that on some level, we're aware of where a robot is standing in position to us, and we react to this. I'd like to show a video that introduces our robot and gives an example of some of the problems we show.
Hello. My name is Nicole. I'm a robotic learning companion. Today, I'm going to be your student, and we'll learn about math. I've learned how to multiply and divide, and I'm learning about ratios now. It might take me a few moments to respond, but don't worry, I am still listening and processing, so please be patient with me. I want to learn how to solve this problem. What do we do next?
Oh, it's just a ratio problem. You just have to multiply.
I am not sure what you mean. Could you give me some hints?
So since we know that Lunara runs 1 kilometer in 6 minutes, to run 3 kilometers, it would take her 3 times longer. So we multiply 3 and 6.
I think I understand now.
Yay.
So we multiply 3 and 6.
Yep, that's great.
The answer is 18. So it takes Lunarin 18 minutes to run 3 kilometers. Is this correct?
Yes, good job.
So this here is a table that students see on the tablet. That was a tablet that you could see in front of my lab mate. Fun fact, my lab mate is actually not that fond of our robot. She thinks it's a little creepy. I personally think our robot Nico is adorable, but that's part of what we're studying. So in this experiment, some of the materials and methods we used. Arguably our most important tool was, of course, Nico, our robot. Nico is about toddler-sized and is capable of dialogue and several movements. We also gave participants a tablet which displayed the problems that they would teach Nico how to respond to. It's a little difficult to see, but in the table there are these blanks, and the blue bar indicates what portion of the problem that Nico and the student are currently on. As the student successfully teaches Nico how to solve part of the problem, Nico is able to fill out parts of this table, and then the student can hit next step when they are ready to move on. Students were given worked out solutions prior to the interaction with our robot, as well as time to familiarize themselves with the material. In addition, we asked our participants to take pre-interaction and post-interaction surveys. There were a variety of questions, but the ones I examined for the study of social formation were statements where we asked participants to rate their levels of agreement on a Likert scale of 1 to 7, 1 being entirely disagree and 7 being entirely agree. So with the experiment, I wasn't entirely honest when I said that Nico was the one filling out the tablet earlier. You see, this experiment, or this project, took place in 2 parts. The part that happened at Mount Holyoke College was conducted in a Wizard of Oz style. What this means is that a human was actually controlling Nico behind the scenes, unbeknownst to the participant. We designed a program that would allow us to give Nico preset dialogue options, as well as controlling Nico's gestures. Because this was a data gathering part of our experiment, we also did have the option to input unique lines of dialogue. Using our data collected from this portion of the experiment, we designed an autonomous program for students to use at the second part of our experiment, which was conducted by our colleague Nicola Leupold at Arizona State University. Now, I'd like to get into detail on some of the questions that we asked and what impacts there were. So we asked 8 questions relating to social formation in the categories of comfort, attention, engagement, motivation, and physical proximity. I'd like to draw your attention to question 1, which reads, I feel comfortable interacting with robots. This was asked in both the pre-survey and the post-survey. Questions 2 through 8 were asked only in the post-survey. So for our results, we found that students in the side-by-side stance did not feel any real changes before or after their interaction with Nico in regards to the statement, I feel comfortable interacting with robots. However, in the face-to-face stance, we found that participants following their interaction with Nico actually reported higher levels of agreement with the statement, I feel comfortable interacting with robots. This may suggest that in the face-to-face stance formation, comfort with robots may increase over time. We did not find any impact on engagement, motivation, or proximity. These are some tables for other results, and I'm going to keep moving, but if anyone's interested, I have copies of my poster still. So in conclusions and in summary, we compared students' self-reported attitudes in comfort, engagement, motivation, and physical proximity across 2 social formations, face-to-face and side-by-side. We found that stances might impact some self-reported attitudes, but not others. And one of the highlights of this experiment was that comfort with robots may increase over time in the face-to-face formation. This is of particular interest to me, as I think it's really cool that something as little as where a robot is standing may impact someone's perception of that robot. And as robots become more and more commonplace in our day-to-day lives, we want to be paying attention to elements such as these so that we can create the most comfortable experience for all users possible. I'd like to express gratitude to all those who made this experiment possible, and I would also like to extend thanks to the audience for giving me their time and attention this afternoon. Thank you.